In this lecture, we will go over the basic principles of Gregor Mendel's theory on inheritance. We'll review some history on how Gregor Mendel was able to be successful in outlining the principles of heredity where other people had failed, and we will start a discussion of the Mendelian laws. I'm sure that many of this material will be familiar to you, having been covered in previous courses. My goal here is to make sure that we're all starting out on the same foundation, and if it's been a few years since we looked at these things, that our terminology and our understanding is fresh. So we're going to go over the basics of heredity, and we'll begin to discuss in this lecture the experiments that Mendel performed and the impact that they had on his thinking on inheritance. So why was Gregor Mendel successful in teasing out heredity where other people were not? Obviously, from the beginning of time, human beings have been manipulating the heredity of organisms to suit their benefit. Animal husbandry is a wonderful example. Agricultural husbandry is another example. If you think about the development of wheat over time to be larger grains, larger heads of grain, so that every plant produces more seeds per stalk. We think about all of this horticulture and agricultural design that we've manipulated over generations, looking for healthier, tastier, larger animals and vegetables and fruits. Human beings obviously had some understanding of heredity. Nevertheless, there was not an overarching theory, structure, viewpoint, or model that explained how heredity worked. And Mendel was successful where others had failed, in large part because of his training as a monk and his strategy. One of the things that helped Mendel tremendously is the experimental model that he used with the pea plants. Pea plants are very useful as a model because they're relatively easy to grow and to maintain. They're very cheap, which is good for someone trying to do science on a monk's budget. They produce a large number of offspring. Their characters are easily discernible, and it's something that he could do on the side because, remember, this was not his primary job. His primary job was his religious duties, and so he did this more as a personal interest or a hobby, we might say. But because he was trained to be methodical, because he was trained to be patient in this monastic lifestyle that he'd adopted for himself, he went far above and beyond what a scientist of his day or even a scientist of our day would do. So the model helped. His approach was very thoughtful and mathematically based, and he was fortunate in the characters that he chose in that they were easily differentiated from each other. One of the things that's interesting to me personally is the times in science where we just got lucky. And Gregor Mendel, in many ways, got lucky in the sense that the characters that he chose to study all followed what we now call a Mendelian pattern. He looked at so many different characters, it's kind of astonishing he didn't find one that had a Mendelian exception. We'll do a whole lecture, 2 or 3 actually, on Mendelian exceptions. And if Mendel had observed some of those exceptions, I don't know if he would have been able to articulate his 2 laws. And so the characters that he chose to study helped him clarify what it was that he was observing. So here's a picture of Gregor Mendel. In his lifetime, he tried to popularize his work. He didn't really get anywhere with it. He was coming from the outside of the scientific community. He wasn't able to make progress in convincing others of what he had discovered. But after his death, these 3 scientists found his work, and they realized how powerful it was, how strong the data was, and they popularized it. And everyone pretty much realized what a game changer it was. And then immediately, across the world, scientists began to try to replicate Mendel's discovery in the models that they were working on to see if his laws were unique to plants or if they were broader than that. And in many ways, they observed the same thing that Mendel observed. But also pretty quickly, we realized that the situation was just more complex, and we began to accumulate exceptions to the two laws that Mendel outlined. So let's do some basic review. Mendel was interested in the transmission of traits and characters. So a heritable feature that varies among individuals is referred to as a character. So flower color, hair color, eye color, height, anything that can be inherited and varied, that's the character. The trait is the specific example. So purple flower, that's a trait. The character is flower color. Peas had several characteristics that Mendel could work with, and there were several advantages to this. As I said, this was a good model for him. They have a short generation time, they produce a large number of offspring, and he could control their mating. Unlike animals, it can be difficult to control mating, especially if you're doing this in a low-tech way, the way that Mendel was doing it. He could control the mating between parents and make sure that he knew the lineage of every pea plant in his garden. So here's a chart of several characters that Mendel looked at: seed color, seed shape, seed coat color, pod shape, and all of these are pretty unambiguous. You can tell the difference between yellow and green. You can tell the difference between constricted and inflated. You can tell the difference between an axial flower or a terminal flower. So these things are pretty binary, which means you're not going to get confounding data where you're like, well, is it this or is it that? You know, is it yellow or is it green? It's pretty obvious which one that you're looking at, which makes the data collection and analysis much easier for Mendel to do. This is a chart of several terms I expect you to be familiar with. I'm not going to read these to you, but this is just here, you know, for you to be able to go back and say, oh, I remember the word locust, but I don't remember exactly what it means. This is a reference for you. There were two types of breeding experiments that Mendel would do. The first is a cross-pollination strategy where the pollen from plant A is used to fertilize the the stigma of plant B. That's a cross-pollination. So you have, so to speak, a mommy plant and a daddy plant. But pea plants are monoecious, and that's a term that will come up in a later lecture, but monoecious means that both the male and female reproductive tissues are in the same organism. And so because pea plants are monoecious, you can mate the pea plant to itself. And so he used both of these strategies to illuminate what was the pattern of transmission. So let's talk briefly about the way he set up the experiments and the results that he observed. The first broad group of experiments that Mendel conducted are what we refer to as his monohybrid cross experiments. And he used these monohybrid cross experiments to build the data set that led him to what we now call Mendel's first law. So after doing this experiment, he came to 4 conclusions that we will follow through the experiment and observe. Now this next section is going to be a little bit unusual because I'm blending in language that Mendel himself used and language we now use in modern genetics. So, you know, Mendel had no idea what a chromosome was. He didn't know what a gene was. He talked a lot about factors. There's a purple factor and a white factor. So I'm blending in some more of our modern terminology, but understand this is not the way that Mendel would have described it, because he didn't have the understanding of cell and molecular biology that we obviously have today. So Mendel first performed a monohybrid cross where he mated 2 pea plants that differed in only one trait. And he created populations of plants that were all the same for this one trait, and he called them true breeding, meaning that he had a population over here of purple flowered pea plants, and whenever he bred them with other purple flowered pea plants or self-bred them, self-crossed them, they always produced purple flowers. And then, in another area of the garden, he had all white, true breeding pea plants. So here we have our parental generation, we have true breeding purple population and a true breeding white population. Now you can also just, appreciate how much work it takes for Mendel to maintain these different populations of pea plants so that he can do these experiments. So he takes a true breeding purple and a true breeding white and he mates them. And you're probably familiar with this experiment. You know that in the first generation, the F1, the familial generation, all of the plants had purple flowers. So I know this is obvious to you and I because we have been, exposed to this information over and over. But it's not obvious to Mendel when he does this, right? One parent is purple, one parent is white, and 100%, without fail, are all purple. I mean, you might even think if you're Mendel, you're thinking, not even one white, not 99% purple and 1% white, they're all purple. And so that was a surprise to him. It's an interesting idea. It's not necessarily what you expect. It seems at this point that one parent is having complete control over what the children look like, and the other parent is having zero control. So if you're thinking this through, maybe it is that there's a purple factor that destroys the white factor, and therefore the purple factor dominates. That could be one interpretation based on the data set we currently have. So he said, okay, well, what I'll do next is I'll take this flower out of this generation and mate it with its siblings, or perhaps I'll mate it with itself in self-pollination. And so he did this several times. So the pure breeding, this is just a review of what I've just previously said. When you cross the two pure breeding plants, they all express one of the parent phenotypes. And one of the things that I mentioned earlier is that Mendel, from his monastic training, is patient and methodical in a way that no modern scientist is. In biology, sort of the standard for experimental repetitions is 3. If you can repeat an experiment 3 times and get statistically similar results, you can go on and submit that for publication, and nobody is going to give you grief. Nobody is going to write back and say, I think you need to do this more. Now, if you don't get statistically similar results, you might do it a 4th time or a 5th time until your p-value is tight enough. But basically 3 is the goal. Mendel did this 60 times. That's just a level of work, commitment, patience that the modern scientist doesn't have really the time to do, because we've got to get the next grant, the next paper, so on and so forth. And so it's one of the benefits that, Mendel, his bills were paid, so to speak, by his monastic work, and he could do this as long as it took. So, he did it once, and he did it again and again, and 60 times he always got the same result. So he says, okay, what happens if we mate the siblings or self one of the offspring? And you're probably aware what happens next. In the second familial generation, F2, he got roughly 3-to-1 purple to white. Now, once again, I know this is not a big deal to you because you've seen it multiple times, but I imagine this is a difficult thing for Mendel to wrap his head around, because the white color of the flower reappears. So as we previously said, maybe if you're looking at just the F1 generation data, you think, well, whatever makes a flower purple, let's call that the purple factor, is destroying the white factor, and so that's why there are no white flowers. But here what we observe is that the white flower is not destroyed, and it's hibernating, it's sleeping, it's laying dormant in the F1 generation, and then it returns in the second generation. The other thing that you'll notice by looking at this diagram is that the purple flowers all the same concentration of purple, the same hue of purple. So when you mix the purple and the white, You don't get a lighter version of purple in the F1 generation. The F1 purples are just as dark as the P generation purples, and the F2 purples as well. They're all just the same color. So when the white reappears, you might expect in this dormancy that the white has been influenced by the purple factor, and maybe instead of pure white, it comes out sort of like a light purple. But it doesn't. So not only does the white phenotype reemerge in the second generation, it's not been destroyed, and it's been completely unchanged. It comes with all of its strength, and it's not been influenced at all by the fact that it was dormant in the first generation. Now, once again, we understand, the rules of dominance and recessivity because Mendel observed them. But this is a surprising thing, I would imagine, if you're looking at the data for the very first time. So once again, this is the review of what I just said. We get this 3-to-1 ratio of one trait to the other, and Mendel, being Mendel, repeated this over and over and over. And then he thought, well, maybe it's just a color thing, right? Maybe this is not universal, it's just the way that color works. And so he went on to look at other characters. Now I want you to take a moment to look at some of these numbers. So he does the flower experiment, and he ends up with 705 pea plants that are purple and 224 that are white, and that's a ratio of 3.15 to 1. So that's almost 1,000 plants. The N value here is almost 1,000 plants. Now, when I'm doing an infectious disease experiment, it's not unheard of in my experiments to have, for every experimental group, maybe an N of 20 or 30 animals, and I may have 3 or 4 experimental groups. So I'm dealing with relatively large numbers of animals in my experiments, but never, even in multi-year experiments I've done with pigs or with mice or with rabbits, have I ever approached anything like 1,000 organisms. Look at the seed color experiment, 6,000 to 2,000. He counted 6,000 yellow peas. And remember, this is not all happening at once. This is through multiple breeding experiments. So I'm just stressing that one of the reasons when Mendel's work was rediscovered that it had the impact that it did was that De Vries and Correns and this team looked at these numbers, and they looked at the statistical similarity, and they looked at just the brute force data, and they were floored by the weight of Mendel's work. And that's one of the reasons why it had the power that it did. Now you'll notice it's about 3-to-1, so we've got 3.15, 2.82. Now in a later lecture, we'll talk about how you use statistical testing to answer the question, 2.82 does not equal 3, but is it sufficiently different from 3 that we care, that it matters? And there's a suite of statistical tests that you can use to answer that question. So it's about 3. We would be very suspicious if it was 3.0 every single time. In fact, some scientific historians accuse Mendel of cherry picking the data because this data is so on the nose, so reliable. I don't have a dog in that particular fight. I don't know if he cheated or not. I tend to think he probably didn't because there's no real reason to. There's no motivation for him to do that. He's not in a publish or perish environment. He's not trying to get tenure. He's not trying to get a grant. There's no reason for him to do it. This is sort of a personal project. But that is a debate in the history of science. Okay, so let's look at this first conclusion. Mendel's first conclusion is one character, flower color, must be encoded by 2 factors. There must be a purple factor and there must be a white factor. And that's because the unobserved phenotype must be present in the F1 generation because it reappears. So the fact that it shows back up means that that white factor is not diluted, it's not destroyed, but is instead hidden. So there must be two factors, and he calls them a purple factor and a white factor. Second conclusion, these two factors, now we call them alleles, separate when the plant produces its gametes. So the idea is that, parent A and parent B have two copies, so in this case, purple and purple, white and white, and when they produce their gametes, eggs or sperm, they separate these two gametes. So there's another egg over here that's capital purple, and there's another egg over here that's lowercase purple. And so they separate, and then the two gametes fuse to create a set of two. Third conclusion. When the factors are in agreement, right, there is no need for a discussion. If they're both purple, then it's purple. If they're both white, then it's white. But there must be this concept of dominance and recessivity. Okay, so the trait that he observed in the first generation, he called the dominant trait, and the trait that disappears is the recessive trait. But that recessive trait is not diluted or destroyed, but is still hidden inside the F1, though it's not active. It's not able to express its will. It's muted. So here in this diagram, we observe that the F1 generation, there is a difference, there's a disagreement in alleles, and therefore one of the alleles makes the decision, and the other allele does not get to contribute to the decision at all, right? It's as if you have two people that are trying to decide what they want to have for lunch, right? One person says, I want to go get pizza. The other person says, I want to go get sushi. They don't compromise and get a pizza that has rice and raw fish on it, okay? They're either going to get pizza or they're going to get sushi, and the dominant person in the conversation is going to completely get their will, and the recessive person in the conversation is not going to get what they want. So if the sushi is the recessive person, then they're going to eat pizza, and they don't get to express their desire or will at all. Now this is a concept that we'll come back to later in Mendelian exceptions, but we refer to this as complete dominance. So Mendel didn't use this term because he didn't know it. He didn't know anything about it. But we refer to this idea as complete dominance, meaning the dominant factor or allele completely controls the decision. Fourth conclusion, the two alleles separate with equal probability. And this is what explains why the Punnett square works the way that it does. The probability of donating one allele or the other allele is even. And that explains the 3-to-1 ratio that Mendel observed. So if we look here at the F2 generation, this plant, call it plant A, this plant we'll call plant B. The genotype of plant A is dominant P, recessive P, and the genotype of plant B is the same. Okay, and so we would call these heterozygotes because their alleles are not in agreement. They're not the same. So the probability, and we're going to get into probability in a future lecture, that they donate this specific allele is 50%. And the probability that you donate this particular allele is also 50%. And it's the same for plant B. This allele has a 50% chance of being donated, and this allele has a 50% chance. And that is why the alleles produce the pattern that they do, because if you think about a Punnett square, right, so if we were to come over here and draw this Punnett square, okay, the probability, if I have 1/2 and 1/2. The probability of this square is 1/4. And if I have 1/2 and 1/2, the probability of this one is 1/4, and this one is 1/4, and this one is 1/4. Now we're going to have a lecture on probability, but if I ask what is the probability that I get purple, you'll add these together, and that is why we get 3/4 and 1/4. So the reason the Punnett square works is because the probability of donating this allele versus this allele is the same, 50% in either direction. This idea is the basis for Mendel's first law. Mendel's first law is sometimes called the law of segregation or the principle of segregation. And the law of segregation says that for every individual diploid organism, they possess 2 alleles for any particular character, and these 2 alleles segregate or separate evenly. The probability that they segregate is even. Now, another concept related to this, but not really one of Mendel's laws, is the concept of dominance. That when the 2 alleles at a present locus or genotype differ, one will make the decision and the other will not contribute to the decision of the phenotype at all. Now, Mendel had no idea why this worked. He couldn't possibly know the mechanism behind what was happening. And I'll tell you as a scientist, this is extremely frustrating because Mendel had no chance of being right because there needed to be another 150 years or 100 years of cell biology research before he could even hope to understand. And so there are things that we think we understand now or we're trying to understand, and the reason that we can't come to a conclusion that matches the data is because there's some nearby field that still needs another 75 years of research before we'll be able to understand what's happening. And so I guess what I'm saying as a scientist, it's frustrating to think I may never be able to have an answer to this question simply because I was born 75 years too early. So Mendel had no idea why this worked. But now we understand that the reason these probabilities work is because of the way that chromosomes are moved around in meiosis 1 and meiosis 2. So this came along later. This is called the chromosomal theory of inheritance. It was put forward by Walter Sutton in the early 1900s. And the idea here is that the genes, the alleles that Mendel observed, are found on the chromosomes. And it is the way that the chromosomes line up in meiosis that explains this difference, right? So if we say, let's ignore crossing over at this point because that complicates things, and we'll come back and talk about that. Let's just take it on its simple fact here, right? If we have in our gametes these 2 alleles, dominant R and recessive R, okay, Mendel says there's a 50% chance that we produce an egg with the dominant R, and there's a 50% chance that we produce an egg with the recessive R. That's what Mendel says. He doesn't know why. That's what his law tells us. So if we go forward in time to now that we understand chromosomes and that alleles are found on chromosomes, we know that in meiosis we're going to duplicate, we're going to synthesize our DNA and duplicate our chromatids. So now I have 2 chromatids with a dominant R and 2 chromatids with a recessive R. And then when I go through meiosis, in meiosis 1, I'm going to separate my homologous pairs so that I end up with a cell that is all dominant R, in this case from my maternal chromosome, and all recessive R's from my paternal chromosome. And when I do meiosis 2 and separate the chromatids, I'll end up with 1/2 of my gametes that have the recessive R and one half that, I'm sorry, the dominant R and one half that have the recessive R. So it is because of the way that the chromosomes move in the cell when we're doing meiosis 1 and meiosis 2 that the law of segregation is true. The movement of chromosomes in meiosis dictates the law of segregation. Mendel had no way of knowing that because he was just too early. We didn't have enough cell biology. But once we figured out what a chromosome was, and we have a whole lecture about this in the future, then people looked and said, aha, it must be the chromosome that is carrying the allele that Mendel observed through experimentation. So walking through this, once again, you've got these two alleles that are found on homologous chromosomes. This one is from mom, this one is from dad. Those two chromosomes duplicate the chromatids, so I now have two copies of the DNA molecule, though still only two chromosomes. We come now to meiosis 1. Once again, ignore this. This is going to complicate things, and we'll deal with that later. When we get done with meiosis 1, we end up with this cell and this cell that gives us our final 1/2 and 1/2 that Mendel predicted. If this is true, and it is, we can use this to do a type of cross that we refer to as the test cross. You can use a test cross to answer the question, is one of the parents homozygous dominant or heterozygous? Now, I will sometimes use the phrase heterozygous dominant. Heterozygous dominant is kind of a silly phrase. It's like ATM machine, because ATM stands for automated teller machine. But I'll use it anyway to really nail down the fact that you can be dominant because you're homozygous or you can be dominant because you're heterozygous. So I know it's a silly thing. I know it sounds ridiculous. I've just learned that it's helpful to frame it so that we're absolutely clear about what we're talking about. So if you have a plant with the dominant phenotype, it could be that that plant is homozygous dominant. It could be that that plant is heterozygous because they have one dominant allele and therefore they're dominant. So in this scenario, we have a plant that is dominant, has a dominant phenotype, and here's a plant with the recessive phenotype. Now in this example, we already know who they are, but this plant could be this, or they could be this. Just looking at the phenotype, they could be homozygous dominant or heterozygous. And so the way that you figure out which they are is by crossing them with a recessive individual. And so in this scenario, because the original parent is heterozygous dominant, we have a phenotypic ratio of 1/2 that are tall and 1/2 that are short. So the function of the test cross is to determine the genotype of the parent who has the dominant. We know their phenotype, they're dominant, but we don't know their genotype. If any of the offspring display the recessive phenotype, the mystery parent must be heterozygous, assuming we're talking about a Mendelian situation. Now on exams, I will let you know, are we in a situation where Mendel's rules are absolute, or are we talking about a scenario where there are exceptions to Mendel's rules? Because sometimes we'll look at it from a purely Mendelian perspective, and sometimes we'll include non-Mendelian situations. And I will make that clear to you in your exams on the questions. Are we talking about a Mendelian scenario or not? So in a Mendelian scenario, if the offspring show the recessive phenotype, the dominant parent must be heterozygous dominant. So once again, this is just showing you the two different ways a test cross could come out. If the parent is homozygous dominant, then all of the offspring will be dominant. If the parent is heterozygous dominant, half will be dominant and half will be recessive. So the test cross is useful when you're looking at a parent and you're saying, okay, I know you're dominant in your phenotype, but I don't know your genotype. Now, when we're looking at an individual who's recessive, their phenotype is known. Two little p's in this case. I hate the letter P as a genetic notation in class when we're working problems and on the exams. I'll much more frequently use the letter A because it's easy to know what a capital A and a lowercase a look like, whereas with P it's difficult to know. That's just a side rant. I hate the use of P as a letter for this, but because it's purple, they use the letter P. That's rant over. Let's move on. So that's the overall part 1 review of Mendel's results. We will move forward in future lectures to examine the dihybrid cross experiments and the implications of those experiments on Mendel's understanding of how heredity works.